Okay, welcome back after lunch. The desserts were over, so I'm guessing everyone liked them. We'll be continuing from where we left off in the morning. And do you guys all have this link? Just show of hands. You've been able to, okay. So it's actually a bunch of tutorials we made last year. Actually it's been made by folks in the lab in the last 5 years and updating it. And finally we got it on Google Colab notebooks last year. So we have 3 tutorials. We'll be going for the first one today. But just to give you a sense for what we'll be covering, and you know, if you do enjoy some of this, you can perhaps go through the remaining tutorials in your own time. And the way it's structured is that in the first tutorial, we'll simulate some genomic data, right? And the idea here is that if you simulate genomic data, then we know what the underlying signal is, what the underlying features are, right? And so when we train models, we can actually see if the models are learning make sense in light of how we've simulated the data, right? And of course, in real life, with real data, of course, we don't know what the underlying features are, what the underlying distribution is. But at least if you work with simulated data and we see that what the models are learning makes sense, we can have some faith that this will also transfer over to real data, right? And so once you train these models, we'll see what these models are learning. That's part of the first notebook. The second notebook is trying to understand, is to interpret these neural networks and see what features at every sequence, for every input sequence, what are the features that these models are paying attention to. So this is a pretty interesting topic, an active research area right now where people are trying to figure out how to interpret DNA, deep learning models. A lot of other speakers also spoke about this, but these tutorials are focused specifically on DNA sequence models, how to make sense of the kinds of features that these DNA sequence models are picking up. And there's a lot of tools, some of them developed by our lab, that have shown good success, especially for genomic data. And in the third tutorial, which is a bit more involved, we actually train models on real data, ChIP-seq data, and then we score DNA variants that are known across people to have different binding of specific transcription factors. So these are SNPs called BQTLs. I'm sorry if the terminology is confusing, but essentially we'll score real variants that DNA sequences that are different between different individuals and see how they differ in terms of binding of transcription factors. Right. So let's sort of get started and open the first notebook. And just before we begin all of this, I think some people had come up to me and asked me how these models are applied in the clinic or even otherwise. So I would say a lot of this work is still pretty basic science research, and much of it is far from like actual clinical use. And so a lot of focus with these models. See, we train these models on experimental data that we've already collected, right? So if you're training a model to predict experimental data, it's kind of pointless because you already have the data, right? But it becomes interesting because often we collect these transcription factor binding or regulatory activity data sets for one individual or for one cell line. But what we really want to do is be able to predict across individuals how this transcription factor will bind in given your genome, right? So once we train these models, we can definitely make predictions on individualized genomes. That's actually the third notebook. So that's one of the use cases for these models, is understanding how your personal DNA sequence will be characterized by these different epigenomic marks. Another interesting use case that I worked on slightly and is sort of up and coming is to design regulatory elements. And the idea here is, if you think about gene therapy, is sometimes we want to deliver a copy of a gene in a very specific cell type to an individual. So let's assume they have some gene that's defective for some reason, or it's just not expressed, and we want to supply them with a working copy of the gene, usually through an AAV construct, which is not that important, but to deliver a gene and some way to control the expression of that gene and make it really cell type specific, right? So as an idea, so when you sort of get your virus shots, the mRNA is basically transcribed in all the cells it reaches, right? And it's not really cell type specific. The only localization comes through how you deliver the virus, right? But often, even when you deliver it, let's say you want to deliver some gene therapy to the eye, cells of the eye, perhaps you don't want to target the cones and rods. Maybe you want to target the retinal ganglion cells, right? How do you do that? And that's become an interesting research topic. And the way people are thinking about it is that once you understand the regulatory logic of DNA sequence, you can start to be able to figure out how to design regulatory sequences that only turn on in specific cell types and they don't turn on in the other cell types, right? And a lot of this work can be done. So imagine like you have cell types A and B, and you have a model that takes in DNA sequence and predicts the activity of that element in cell type A and cell type B. And let's say you want to design a sequence now that is high in cell type A, has high activity in cell type A and low or no activity in cell type B. The way we do that is, let's say we start off with a random sequence and we start making some edits to that sequence. And then we predict, the predicted the activity of this sequence in both cell types. And we keep making updates so that the prediction as per the first model increases, and for the second model either decreases or stays constant. And if you keep making these updates sort of in a greedy fashion to a starting random sequence, you can end basically with design sequences that have desired properties, so desired regulatory activity properties. So that's a pretty interesting area of research that perhaps will have some clinical relevance in the next few years. Okay, so let's do this. I hope you've opened up this notebook. Yeah, cool. So it's pretty self-explanatory. There's a lot of text, so let's go through it part by part. I think the way we should work through it is, I'll not say a lot of stuff. You guys should read, take your own time. Of course, I'll jump in from time to time. Raise your hands and ask questions as and when you have any. And of course, please ask everyone so we can discuss that, and so that everyone is on the same page. So I'll give you a high-level overview of what we're doing in this notebook, and you can start running cells. So I hope you have familiarity running a Colab node, right? So the first thing I would want you to do is, if you go to runtime and you click on change runtime type, just make sure that this GPU is selected for you. So what this does is, since we'll be training some machine learning models, running them on a GPU will make it much faster. So is that good for all of you? If it's not, you can just select GPU and then click save. So, if you read some of these things, that's basically doing what I mentioned. And what we'll start off with is just installing some required code. So the way you run a cell in Colab Notebook is either you can click it here, or you can press Shift and Enter, or Shift and Return on a Mac. That's usually what I do. It'll ask you if you really want to run it, and you can, if you trust me. And then you can install these packages. One of them is called the DragoNN package. It's not really well maintained by a lab, but it's useful. It's pretty relevant for these tutorials, so definitely check those out. And then we start off with some imports. We're importing NumPy, we're importing TensorFlow, which we'll use, matplotlib for plotting. And as you run those cells, you can, the install will take some time, so we can keep discussing and chatting about DNA sequence and these models. And so if you remember this from the morning, DNA sequence usually has these DNA words that are recognized by transcription factors. And often this recognition is kind of soft in that certain mismatches are allowed at specific positions. And there's basically this spectrum of binding affinity between DNA and transcription factors. And transcription factor sites typically have motifs, multiple motifs, which can often be of the same transcription factor, but more often they belong to different transcription factor families. And often it's things like distance between these motifs, the identity of the motifs, how many counts of each motif that you have, that determine the regulatory activity at that sequence. So what we're going to do today is an interesting simulation, right? And as I mentioned, the interesting thing about the simulation is that you know exactly what's going in, so that when you train the model, you can see if what you've learned with the model makes sense, right? Because you're controlling the whole process. And so what we're going to do is simulate some data, right? We're going to simulate positives and negatives. It's a binary selection. It's a binary prediction task. And so the way we're going to do this is we have this motif that's called TAL1. Again, not very important, but you can see that it looks like this. So it prefers a CCA, and then it kind of is okay with everything, and then a CTG, right? And so what we're going to do is, in our positive set, we're working with sequences of length 1000, right? And these sequences have a certain composition, GC composition. So basically they'll have some fraction of G's, some fraction of C's that we predetermine. Again, we're simulating the whole thing. And then we insert instances of this motif in the sequence, right? And the way we're doing it is that in our positive set, we're inserting up to between 2 to 4 instances of this motif, but in the central 150 bases, right? So any example where there's 2 to 4 instances of this motif in the middle 150 out of the 1000 bases, we call that a positive example. We're simulating all of this. So this is not super realistic, but it'll give you some sense for like how these models combine not just sequence patterns, but also positional arrangement of these motifs. And in the negative sequence, negative sequences will also contain instances of this TAL1 motif, but they can be spread out. So they don't have to be necessarily located in the central 150 bases, right? Does that make sense? So no reverse complements. We're just keeping it very simple and so that people understand. Well, we'll look at the model weights and see what's, so not super realistic, but a good starting point. Yeah. No, so it's in the absolute 150 bases in the center. Exactly. So anywhere but in the central 150 bases. Yeah, I think it's like a uniform number between 2, 3, and 4. Yeah, yeah. Okay, so you don't have to do the simulation yourself. It's all kind of done for you guys. If you just run some of these code blocks. Once you run this one, actually, it just gets some simulation data for you. It also splits them into training, validation, and tests. So are you guys familiar with train, validation, test splits? Can you just raise your hands? Anyone who's a bit confused about it? Okay, cool. So we're great. That's great. So what this is saying is we're going to simulate this set of sequences. We'll generate 3000 positives and 3000 negatives. And it's saying that the GC fraction is 0.4. That means in our sequence, of course, we have to generate some sequence that's 1000 long, right? And so the way we generate is that we randomly sample so that the probability for G is 0.2, of a C is 0.2, and then of an A and T are 0.3 each, right? That's just a choice that you're making. And then it's going to insert instances of this TAL1 motif in sequences of length 1000. For positive examples, it's going to be in the central 150 bases. Yeah, so that's going to be our data. And you can actually just run the next few cells. And mostly what these cells are doing is preparing your training, test, validation matrices, arranging all of these sequences into these splits, and also arranging the labels. So what you can see here, for example, is that you have the training data. So the X for the training data are 4000 examples. Roughly, it'll be split between positives and negatives. You have the sequence of length 1000, and then you have 4 channels. And these 4 channels correspond to AC, G, and T. So if you remember the one-hot encoding that we had spoken about in the morning, that's what it looks like, right? So you have to convert every DNA sequence into this sort of one-hot representation, right? That's because these models work with matrices as input, not sequences. And so again, you don't have to do this by yourself, but what's going on is if your input sequence looks something like that, it's converted automatically into a matrix that at every column has a value of 1. Yeah. On Colab? You can, well, till a few months back, they would let you run 3-4 notebooks in parallel, but now they become very restrictive. Oh, yeah. So yeah, I think you can often split a GPU into like multiple models depending on how much memory is being used. But often these models take up most of the memory, so you can really use one GPU only for one task at a time, for one model training at a time. But there are some larger GPUs that have like 80 GB memory now. Yeah. So if you have these A100 GPUs, they have a memory of 80 GB now. So you can just train multiple jobs on that at the same time. Yeah. Yeah. So I mean, so if you do A equals 0 and that's basically ordinal. So it essentially suggests that there is a link between, the magnitude. And so basically, because it suggests that, so if A is 0 and C is 1 and G is 2 and, and 3, so that suggests G and T are closer to each other than A and T, right? But that isn't usually a choice. Right, yeah. The number of rows in this case, yeah. If you have a longer vocabulary, if you have like for proteins, you'll have 20 rows. But this is usually the most canonical. Even if you look at natural language, for example, people used to work with words as their vocabulary size, and they'd use like 100,000 words. So this matrix will have 100,000 rows. Now people have moved towards some different encodings, but it's the same idea. Right. So there's some interesting, there's a lot of data processing happening here. We're basically all, we're just arranging all of these matrices into train, test, and validation, as I mentioned. If you reach this cell, when you do, it's going to show you, we've actually written out these sequences to a file. And this file is called a FASTA file. So if you ever work with any genomics data, you'll probably come across a FASTA file. And it's a very, very straightforward file format. As you can see, it's just got these greater than signs. That's demarcating. So that's basically giving you the ability to name each read. And so you can essentially write, yeah, exactly. It's the header for that specific sequence. And then you have the sequence itself in the next line. And then you have the header for the second sequence. So the label for the second sequence. And the sequence corresponding to that. So often if you work with any genomic data, you probably see a file format like this. But all this is really doing is the matrices we have generated with simulation data, it's writing them to a file. If you're working with your own real data, you'd probably have to just swap in this file with a FASTA file that has the sequences from your experiment, right? So if you want to run this on your data and you have through some experiment or through some other source, you've been able to get a positive set of sequences and negative set of sequences. All you have to do is put them in a FASTA file, and then you can probably just swap out the simulated data with your files and just run this notebook from there. Right, so you keep running, and if you reach this point, just raise your hand and let me know, and then we can go on from there. And if you have any questions, feel free to ask. So I'll just wait maybe 2 or 3 minutes till all of you run cells up to here. And just one thing to know is that the labels here are in this Y train and Y valid and Y test. And these are just vectors, right? And they're just binary vectors. So the labels correspond to the positive examples. So the examples where these sequences are in the central 150 bases, they'll have a label of 1 and 0 otherwise. So when these motifs are scattered everywhere in the sequence, the label associated with that will be 0. Can any of you think of a reason why these labels might not always be correct, even though we have simulated this data and assigned these labels? Or given the way we have simulated this data? Mm-hmm. Yeah. Yeah. Yeah, that's perfect. So that's one possibility, right? So the negatives, we kind of randomly select the start sites, but there can be cases where just by chance. That's great. Another thing I was thinking about, does anyone have any other ideas? Yeah. Yes. So yeah. So great point. So I showed you as the motif that we're inserting in these sequences look like the CCA CTG thingy, and of course it's sampled at random at every position, and sometimes you might just choose sort of a very weak instance by chance. Right? Yeah. Yeah. So exactly. So in this case, it's a bit less likely, but if you work with most real, well, this is a real motif as well, but if you work with some other motifs, this can happen. Any other guesses? So one thing I was thinking of is that the background sequences, these are like sampled every position from the GG, from the uniform distribution where, not uniform, where from a distribution where G's and C's have a 0.2 probability and A's and T's have a 0.3 probability. So you will have some cases where this, even in your background, this motif just pops up by chance, right? Because even if you sample every position from a distribution, from a given sort of distribution with fixed weights for GC, which have fixed probabilities for AC, G, and T, there is a probability that there's a good chance that from all of these 3,000 sequences, each of length 1,000, some of them just by chance have this string CCA, CTG. Yeah, I think, yeah. Yeah, the label, if you place them inside, yeah. So I think you're right in that you can't have false positives. Uh, yeah. 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 I think no. Yeah, maybe not. Uh-huh. Yes. Yes. Uh-huh. Uh-huh. Yes. Yes. Correct. But we have 3,000 sequences. So maybe you might see, it's true that you might not see a lot of them, but for a shorter motif, you're right. You're right. It'll just be the sum across. Yeah. Yeah. Well, so let's say 1 by 4 to the 7, right? So it's 1 by 2 to 14. That's 1 by 8,000 or something, right? Or 1 by 16,000, right? So at every position, it's like a 1 by 16,000 chance. And you have 3,000 times 1,000 positions, right? 3,000 positions, 3,000 sequences and 1,000 positions. So 3 million. So you'll have some expected number, right? Sorry? That's true. Yeah, I think we were just talking about one specific insertion of one specific element by chance. If for all three, right? Yeah, yeah, correct. But you can have something that's labeled positive, but has by chance one element outside. That can be labeled potentially. Great. So has everyone reached this point? Show of hands. Anyone who's not reached that point or had any issues running the cell? Awesome. Okay. Doing great. So, okay, we've got the data set up, I think, which is usually the hardest problem because often this, if you're working with real data, it'll come from, you'll have to wait like 3 months to actually get it sequenced and then maybe a day to actually like process the raw sequencing reads and then finally come up with this matrix. So, right, so we ran through all of this. So the next thing you need to do is set up the model, right? And the way I like to think about it is if you use a framework like Keras or PyTorch even, it's a lot like Lego, right? You have all of these layers and you have to figure out what the right model to build with them is. You have to figure out how to fit the fully connected layer with the convolution layers, with now the transformer blocks. And so you can do this in very creative ways, but often like if you look at Lego, there's some structure, right? People usually use certain building blocks, and there are some sort of conserved themes, some certain patterns that emerge more often than not, right? So similarly, you'll often use a fully connected layer followed by a nonlinearity and another fully connected layer. And in sequences, or even for vision, you'd use convolution layers followed by max poles, convolution layers, max poles. You'd flatten the output at some point, and then run fully connected layers on top of that, right? So that's the general idea. And so we'll start off with a very simple model, right? It's still non-linear. So if you remember what we'd done in the morning, we tried a one-layer convolution network, right? With just one filter. And we'll take sort of the maximum output. We'll do a max pooling on top of it, and then combine all of those to get one prediction, to get a positive or a negative classification, right, prediction. So we're going to set this up. So this is what the model is going to look like. Maybe not the best cartoon, but let's see if we can explain that. So we'll start off with our input sequence. Okay, so this is we're designing a model, okay, and we're designing a model which just has one convolution filter and one fully connected layer, and in between that there's one max pool layer. The reason this is interesting is not because it's the best model that we can do, but it's something that we can train and then inspect to see if the model through the learning process is learning the right motifs, is learning the right rules, right? And so what we'll do is start off with the input sequence. And then we'll have a convolution layer that we'll scan across the input sequence, and that'll give us one value per position, right? Barring some edge cases, because as your model slides, the width of the convolution will cause you to have fewer outputs than positions in the input. Let's ignore that now. So you have one output at every position, and then you can threshold that using the ReLU nonlinearity that we had mentioned. So everything below 0 gets clamped to 0. And everything above remains as is, right? And then we can take the maximum. Perhaps in this case, it's showing you the maximum across the entire sequence. But so what ends up happening in the real model, which is the one on the right, is you take max pool of every n positions, right? And then you have sort of a fully connected layer, and that makes the final prediction, right? So this is the, okay, this is the full model, right? So let's see, how's this working? Right. So this is showing you the case of a more general model, which has more than one convolution layer, right? So again, you have your one hot input sequence. In this case, you have two filters. So let's say we'll start off with a one filter model, and then we'll add more filters. But just for generality's sake, let's consider the case where we have these two filters. Initially, these will be randomly initialized in the beginning, and so they'll actually look pretty noisy, something like this. And Google Colab keeps reminding you that you should use. So what you're going to do is scan both of these. So what the model will do is scan each of these filters along the sequence. And so what you'll get is two outputs, right, at every position that'll correspond to the math score between the sequence the subsequence starting at that position and each of these convolution filters, right? You get two outputs per position, right? And then you apply some nonlinearity, which will be ReLU in this case. And this is an element-wise nonlinearity. That means it really applies independently to each element. Here, I think the blues are negative, the reds are positive in this cartoon. So you can see everything that's blue is set to 0. Everything that's positive retains its exact value, and it's kind of washed out, right? Okay, so this is showing you a model with two convolution layers. So you can actually repeat this process, have more convolutions, at the end of the first layer, more convolutions, come up with, in the next layer, you'll have 3 outputs because you have 3 convolution layers here. And you can kind of repeat this process, do a ReLU again. And then this is what gives you sort of the output of your convolutions. And when we apply a max pool in this case, so here we are doing a max pool of size 6 and stride of 6. What that really means is that for every 6 positions, we're taking the max per channel. Each of these is a channel of the filter. So the way, again, this looks is that we take a max along this, and that gives you this value. So I think it corresponds to this one, right? And then you do a max along here. I think this is the max that shows up here. You do a max along this channel. You see it's lower, it's this one that shows up there. And that gives you basically this one column of your max pool, right? So what it's doing is you're losing some resolution, right? Because you're compressing the information of 6 positions into 1 position. You definitely lose some information, but what it's doing is it's trying to aggregate information across positions. And if you're interested only in whether the sequence is bound or not, that's usually okay, right? Or whether it has certain motifs in certain positions, right? So like going back to our simulation, we're interested in whether the motif is present in the central 150 bases, right? So it doesn't really matter if it's present in from positions 500 to 550 versus whether it's present in the entire central 150. So you're okay to lose that resolution. It's all right if you lose that resolution at least in this example, right? And so we repeat this process for every block in this max pool. That gives you the second column, the third column, and the fourth column. And then that's what your max pool output looks like. So, and all of these are, you guys have looked at convolutions for images. So the thing with images is that that there's an extra dimension, right? So there's height, width, and then there's a number of channels. This is a bit simpler in that you just have the length dimension, and the number of channels here is just 4, which is the number of characters, right? So in comparison to an image, which is 3D, like it's a 3D tensor, the input here is just a matrix. It's a 2-dimensional matrix. And so these convolutions are also have 1 dimension less than the dimensions that are used for, than the convolutions used for vision, right? Actually, they're just matrices here. So you do the max pool, and then we can sort of flatten these out. So you have these 12 values. You can just really arrange them 1, 2, 3, 4, 5, 6, 7, 8, 9, and 12 in one single vector. And then you can use a linear unit, so just a matrix multiplication that gives you one final output. And you can use the logistic function that gives you a probability distribution. This nonlinearity will map your prediction to a probability value between 0 and 1. Right? So that's a more complicated architecture than we'll start out with. What we'll start out with is just one convolution and just one filter, and just one layer instead of 2 layers, and then one max pool, and then a linear unit. So let's see if that's drawn out here. If not, I can just draw that out on the board. All right. Maybe I'll draw it on the board. So again, so the architecture in the next part, scroll down to the cell, is what we're calling a one filter model, right? And so the way it'll work is, I'm going to draw it top down, is you start off with the sequence, right? It's the one hot sequence. So it'll look something like this, and it'll be one when there's, let's say, A, C, G, and T. So if your actual sequence is something like A, G, and something, it'll look like this, have zeros elsewhere, and so on. And then what we do is we'll just use a single convolution layer. So it'll look something like this, I think, in our example. So actually, let's start going on the architecture, and then I'll draw it out. So this is how we describe the model architecture in Keras. And it's quite simple if you think about it. So what we're doing is we first say that it's a kind of model that's sequential. All that means is we're going to add layers one by one. And that's exactly what we do. So the next line onwards, we start adding layers one by one, right? So what we're adding is a convolution layer of a certain size. Here you can see that the kernel size is 10. I'll show you what that means. We have this thing called a batch normalization layer. You can ignore that for now, but all it does is some sort of rescaling of the neuron weights. And it's a linear rescaling. And then we add this ReLU activation. We perform max pooling, we flatten it out, and then we use a dense layer and then a sigmoid activation, right? That's a simple architecture we're going with. So, okay, so I'll get back to the drawing board. And so the first convolution filter that we're using has a width of 10. And that's really going to go across this entire sequence. And once you convolve these two, what you end up with is one value for every position in the input. So you're going to end up with one simple vector, right? And what we do next is that we apply a max pool layer. Well, I guess we apply a batch normalization, so you can just ignore that. And then we apply a ReLU activation. So when we apply ReLU to this, as you've seen before, the negatives, so if something is a -1, it gets clamped to a 0. Anything that's positive stays positive, right? And that happens at every element. Yeah. So we can just kind of draw that out. What we're going to do next is this max pool layer. And you can see that the max pool size says 35, right? So if you remember, this sequence has a length of 1000 in our simulated data. And we sort of chunk that out into bins of size 35, right? So we look at 35. So the first 35, the next 35, and so on. And then we take the max across each of these bins. So that gives us slightly fewer than about 30 bins after we take the max pool from this, right? So when we do the max pool, we look at the max for the first 35 positions, the next 35 positions, and what that'll end up giving us is about 30 values, about 1000 divided by 35 values. And again, that's also going to look like this vector, right? What we're going to do after that is just use a fully connected layer. So what's represented by the dense one here, all that means is that we're going to do a linear regression actually. So we just take all of these values and learn a matrix that just gives us one output here. So we're just going to learn a simple function that's really just a dot product with a weight matrix of this vector. And for the final output, we'll also just use the sigmoid probability function, the sigmoid activation function that's going to transform that into a probability that goes from 0 to 1. Okay, so that's the architecture we'll be working with. Does that make sense? Yeah. 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 Great question. So the way it happens is that when you convolve the convolution filter across the sequence, at every position, you'll get the score for the entire subsequence starting at that position, right? So if a motif spans, it'll just sit in one of them, basically, where it starts. So if it starts, so it's on the left one, basically. So we should also look at the number of parameters we're talking about here, right? So what are the parameters that we are learning? So this convolution filter is something that we're going to learn, right? We'll actually even visualize it because eventually it should match the motif that we've simulated in our data, right? So we insert the tal1 motif that you remember. And when we initialize this matrix, it looks nothing like that tal1 motif. But we hope that once we train our model, it picks it up. We'll actually see that. So that's pretty cool. But this has, so this has 40 parameters, right? I'm ignoring some extra ones, but the main ones in this matrix are 40 parameters. We're going to learn 10. So the width of this convolution filter is 10. And the number of channels are 4. So there's 40 parameters that we're learning here. If you remember, in max pool, there's no parameters, right? The ReLU, no parameters, because it's just an element-wise operation, no parameters involved. Again, max pool has no parameters involved because we're just taking the max of every 35 values. There are parameters involved in the final dense layer because it's just a linear regression, right? So we'll have, I think, about 1000 by 35 neurons here. So let's just say that's about 30, right? So we have about 30 here, and then we have one final output here. So we need 30 more parameters to take a dot product of these values and get you one final output, right? So we have about 30 parameters here. Does that make sense? So we have so total we have about 40 plus 30 parameters, and then we have some extras, 2 or 3 bias parameters. So we'll be really learning these 40 odd parameters and fitting them. And so you can actually just run this cell. And what that's going to do is set up the model architecture. We still haven't trained anything. It's completely randomly initialized. Maybe I missed some cells. Yeah, make sure you run all the cells in between. So this is just, what this is doing is it's just importing a bunch of things in Keras. It's importing things like the dense layer, the activation layer, the convolution layer, which are already implemented in Keras. So yeah, as I mentioned, we have about 70 odd parameters that we're training. So you can see that 40 parameters come from here. So actually there's one more parameter that I didn't tell you. So if you do a linear regression, you remember the bias term, right? So if you have a linear regression where your input vector has dimension 10, you'll have 11 parameters. So 10 weights and then the one bias weight parameter, right? So that's where it's actually 30 plus 1 here and 40 plus 1 here. And so that's what it says. We have about 72 trainable parameters, which is the number we're talking about, right? So we have set up the model, and we have to tell the model what the loss function is. We have to tell it what optimizer needs to be used, right? These are typically important decisions, but most of the machine learning community, especially for the optimizer, has converged to an optimizer called ADAM. So it's a bit more involved than simple stochastic gradient descent because it takes into account sort of the momentum of the gradients. So basically keeps some notion of history. And you can try SGD as well, but we just roll with ADAM. And for the loss, since we're just doing simple binary classification, we're outputting a probability, we'll be using the binary cross entropy loss, right? We just specify all of that and compile the model. And we'll be using the AUPRC metric. I think Anshul mentioned the AUC, AUROC, and AUPRC metrics yesterday. So this is just a way to specify what metrics we'll be using and how we'll be computing them. So is everyone here? Show of hands. Any questions? Are we clear on the number of parameters, the model architecture for this simple model? Yeah. Yeah, I think the non-trainable parameters. I think those come from the batch normalization layer, if I'm not wrong. The batch normalization layer, I guess we just ignore that for now. One thing it does is takes a running average across batches. It's just a normalization step. We'll just ignore that for now. The next thing we'll do is, before we even start training is, let's just look at the parameters, right? We've obviously we initialize them randomly. It'll be really suspicious if the model already is initialized with the TAL1 motif that we simulate the data with, right? That shouldn't happen. So why don't you just run these cells and you can start seeing some interesting patterns. So if you look at this, these weights, what these weights actually are the weights to which this linear layer has been initialized, right? The way we are now think about how we simulated the data, right? We have put in the TAL1 motifs in the central 150 bases in the positive examples. And so we should hope that these parameters that are learned have some positional preference, right? Because ultimately you only want to count the motifs that show up in the center, right? So if you look at the weights corresponding to each position in this linear regression layer, I think there's about 27 of them, right? So close to 30. So you can see that it seems pretty random, right? All these parameters that have been learned seem to be just sampled from some normal distribution, probably centered at 0, right? That's what we expect because we haven't, these are completely randomly initialized. Right? And then if you look at the convolution parameters, so this is the motif that we had inserted in, this is the motif we had inserted in the simulation data. And this is the matrix corresponding to the convolution filter that's been initialized, right? So you can see, I can't find the CCA CTG motif. Can anyone spot it? Sorry? Yeah. So it seems pretty reasonably random to me. And I can't find but if anyone cites it, point it out. I can see the GTG, but not much of the CCA here, having the same sign at least. All right, so we're getting pretty ready for training these models. Yeah, questions? Yeah, I mean, the weights are just sampled from some normal distribution, probably centered at 0. So this is just a visualization, right? So it's just that's the value, say, corresponding, there's 4 columns there. I'm just going to expand this out. So it's a matrix that looks something like this, right? 10 positions and then 4 columns, right? And all of these values are initialized randomly, right? Probably centered at 0, I think that's the default. And so some of these can just be negative 0.2 or something like that. And here this value will correspond to the C. So you might see a negative C. That's just how we visualize it. So is this okay so far? We have set up our training data. In this case, of course, we simulated it. So that's fine. So we know what to expect when we train this model. We have set up the model architecture, right? That's the most one of the most important tasks. We have set up the model architecture. We made it very simple because there's a tutorial, right? And of course, we'll look at more complicated models later. And then we've decided what the loss function is. Again, a very important piece. Since it's binary classification, we have set that to binary cross entropy. And then we chose an optimizer. So we used Adam as our optimizer. You can also try using something like SGD if you wish to. So what we'll do next is call this .fit function, right? So we've set up this one filter model. We've told it the loss. We've told it the optimizer, right? Now we need to supply it data. So we'll only train on the training data. We have some validation data. So just a split of our data that will help us figure out when to stop training, right? And then another useful hyperparameter that people use is something called batch size. So are people familiar with batch size? So the idea is that we have 3,000 examples, right? What you could do is compute the loss across all of those 3,000 examples, compute the gradient and then update your model, right? So that's actually what is done in gradient descent. But in sort of stochastic gradient descent or batch gradient descent, just to make this a lot faster, even though the gradient updates are slightly noisier, what you can do is just compute the loss function on, in this case, 128 examples. Use that to compute your gradient and then update your weights, right? So it just so ends up happening that, I mean, there are good reasons behind this as well, but this ends up speeding the process because imagine like computing the loss on all your examples just to make one update to your model, right? That ends up being quite slow. So batch stochastic, batch gradient descent is a good way to compromise between having reasonable gradient updates while also being reasonably fast. So that's what the batch size is used for. Epochs usually tells you how many epochs, so how many passes through the data you want to make till you stop your model. But this is sort of like an upper limit. Often what people do is something called early stopping. So has anyone heard of early stopping? Great. Yeah. So idea behind early stopping is you track the performance, not just on your training set, but also on your validation set. What it's saying is that we don't want our model to overfit just to the data in our training set, right? And the way we monitor this is we don't train, you don't make any gradient updates using our validation data. But at the end of each epoch, we just evaluate, either we look at the loss or we look at a performance metric on our validation data. And we say that once the loss starts plateauing, once the validation performance metric or loss starts plateauing, let's stop training, right? It's possible that you're doing even better on your training data, but somehow on your validation, which is like a held out set, your performance is saturating, right? So it's saying that perhaps this is when I'm starting to overfit on my training data and maybe I should just stop training further, right? So that's the logic behind early stopping. This patience parameter essentially controls how long you're willing to wait till you see an improvement in your validation performance. So if I go for 3 epochs and I don't see any improvement in my validation performance, we stop training. But maybe on the third epoch after, let's say I see the best performance, next epoch is worse, the one after that is worse, the third one is better than the first one, and then I continue training till I see like 3 continuous rounds of no improvement in the model performance. All right, any other questions? We can actually just start training and this can take, I think it'll take a minute or 2 minutes. And often what a lot of machine learning people do is they start training their models. Models can take days. So they just watch these model losses go down epoch by epoch. So often each epoch, if you're working with reasonable sized data, can take hours. And so machine learning researchers have nothing better to do than to just stare at these numbers and make sure that the model loss is going down. So you can see again, this is spitting out a lot of numbers. And just to sort of simplify that for you, is going epoch by epoch, right? So the first epoch, it's seeing the data, it's doing one pass of the data, or the data. And it's giving you a lot of metrics. So it's showing you AUROC, AUPRC, showing you the accuracy. So remember, it's balanced data. There's an equal number of positives and negatives. So it's showing you that number on the training data at the end of the epoch, and also on the validation data, right? So you can see after the first epoch, performance is pretty close to random. It's about 50%, even as the same for your accuracy, for your AURC, AUPRC, they all look pretty much the same, right? And just showing you how many positives and negatives there are in the validation set. So you can see it's almost 50-50, but of course there's some sampling. Because of the sampling, it's not perfectly 50-50. And finally, it computes the cross entropy loss on your training set, your validation set. And this is really the starting point, right? And you can see epoch over epoch, it starts to slowly improve. So it's going down 70-81, 70-80, 70-88. So it didn't improve this time. On the validation set, you can see went down to 70-70, right? So you can keep staring at these numbers. Again, 70-99 is not the best, but I think now it starts dropping. It's 70-65, 70-31, and then it starts going down, right? So as this validation loss is going down, and the loss is something you're minimizing in this case, you can see that the performance also starts improving, right? So you can see this number, this AUPR, that was initially about 0.496 or 0.52. Across epochs, it's starting to improve, and it's going up to 0.58 now. And as you keep running it, actually, I think, yeah, so training is converged in this case. So we can really scroll down to the bottom of this training log. Oops, scroll too much. Where are we at? Yeah. So you can see by the end of training, or close to the end of training, the AUPR has gone up almost to 0.96. So and the accuracy again is pretty high. It's like 90%, right? So even with a very simple model, because the data generation process is pretty simple, right? So if you think about how we simulated the data, in theory, it's pretty straightforward, right? It's inserting motifs and then figuring out where they're placed. And you can see a model like this converges after about 50 epochs and gives you a performance of about 90%. So not too bad, right? For a model that's simple and just 70 parameters, right? But it's thoughtful, right? We thought about how we insert the motifs, and we created a model that sort of matches that logic. So scans with motifs and then counts those motif elements and how many of them are present in a certain area. So were people able to converge the same numbers? Or is there someone whose models are doing better than this or worse? I think it should be reproducible because we've set seeds and stuff like that. But let me know when your training is converged. Has training converged for anyone? Yeah. Yes, few of you. So let's take some more questions. So do these numbers make sense? There's a lot of numbers going on, but the essence is the loss should go down and the performance metrics should go up in general, right? And so long as that is happening, after every round of us seeing the data, that means we're making reasonable progress and our model is improving. Yeah. Yes. So no constraints, right? No constraints at all. This is just a weight matrix, unconstrained. So it can be really anything. We're just initializing it with I think like a Gaussian centered at 0. So some of them end up negative. Can be anything, right? So we'll see that. Yeah. Yeah. So are people familiar with like precision, recall, AUPR, and those kinds of metrics? Anyone know when you should use AUPRC versus something like AUROC or something? Yeah. Right. Yeah, exactly. So What's the, if you have a random classifier, what's the AUC of that going to be? Less than 0.5? Any other guesses? Sorry? Yeah, 0.5, so around 0.5. If you have a random classifier, do you know what the AU PRC of that will be? 0.2? 0.5? Any other guesses? 0.5? AUPRC, not the AUROC. The AUC will be 0.5, but the area under the precision recall curve for a random classifier. 1 for a random classifier. Okay, any other guesses? Same as? So it's not 0.5, yeah. So it turns out the AUPRC of a random classifier is the proportion of the minority class. So if you have one positive to 9 negatives, the AUPRC of a random classifier will be 0.1, the proportion of the minority class. Yeah, we can, if anyone's interested, we can work out why that's the case. And so for a random classifier, you should expect an AUC of 0.5, so an area under the receiver operating curve of 0.5. The area under the precision recall curve will be close to the imbalance fraction of positives in that data set. Okay, has everyone trained their models? Anyone got a model better than this one or worse? Okay, so we have trained the model. You know what is often done is people make predictions on test data, right? So test data is data that's completely held out, and the hope is if the test data is not very different from your training and validation data, that you will get decent performance also on your test data, right? So these are examples the model has not seen either in the training or the validation sets, right? And so if you run that cell, the model is going to make predictions on the test sequences. And then just let's see how the model is doing. And also run the next few cells, and it's just going to show you the learning curve, right? And I'll give you a sense for how we choose when to stop training the model. Okay. So you can see that on the test set, our model performance is also close to the performance on the training and validation sets, which was about 90 point something percent. And you can see also in the test set, it's not much worse than that. So that's a good sign in general. And we can also visualize the training curves, right? So as the epochs passed, how did our model's loss go down? And you can see, so there's actually like websites where people just document their loss functions because loss functions can often have really crazy behavior. So what we're seeing is that initially we start off learning a bit slowly. And then yes, after about 10 epochs, the model kicks in. You can see the loss goes down a good amount. And then by the time we reach about 50 epochs, you can see that the validation loss starts converging slightly. In fact, I think this is the point with the best validation loss. And that's the one we select. And so often people have seen really crazy loss functions that like stay flat for 50 epochs and then suddenly just go down. And there's a whole website with just people posting loss functions over time. So are these things clear? Things like early stopping, tracking the validation loss, why we're even using a validation set. All right, so we have about 25 minutes, which is good because what we're going to do next is look at the parameters that the model has converged on after training, right? So so we have two sets of parameters that we're really concerned about. The first is the convolution layer. So these 40 parameters, and the next is this fully connected layer, right? So does anyone have thoughts about what these parameters should look like for both of them or one of them? Yeah, any guesses? assuming now that we know that we have a model that's getting you an accuracy of close to 90% on this simulated example where our motifs, the example is positive where when the motifs are in the central 150 basis. Any guesses? So let's talk about the convolution filters, right? I think we've discussed this and sort of alluded to the fact that we should expect this to look like something, and people have thoughts on what it's going to look like. Yeah. So we initialized it and it looked like a normal distribution, right? But yeah, is this going to look anything like the motif we had implanted? Should we expect that to be the case, or? So it should look something like the motif, right? Maybe not identical to it, but at least have the CCA, CTG thing. Yeah, yeah. That's a good guess. I think that is actually what ends up happening. We'll even see that. Before that, so let's assume that this this convolution ends up converging to the TAL1 motif that we'd inserted. So what this is really doing is then at every position, it's going to compare the match of that subsequence to the TAL1 motif, right? The simulated motif you'd inserted. Now, does anyone have any guesses as to what these weights are going to look like? Are they going to have some structure to them that we can expect based on the fact that in the positive examples, the motifs are usually concentrated in the middle? So just to sort of jog your memory, once we slide this convolution filter across, we get a score at every position, right? So if there's a match at this position, you'll have a high score here. Then you do a ReLU, which is just going to sort of amplify. It's going to damp out the negative values and keep the high values. Then you take a max pool at every 35 positions, right? So if there's a motif here, it's still going to show up as a high value here, right? So each of these corresponds to 35 base windows, right? So this is going to be high if there's a motif in the first 35 bases. This one's going to be high if there's a motif in position 70 to 105, right? So given this, what do you think these weights are going to look like, right? So in the final layer, we're just doing a dot product of this matrix, which tells you where the motifs are present with a learned weight vector, so 30 values. That's going to give us the final combined output. So can you think about how we can combine this vector, which contains information about where motif matches are found, with what we want in our output, which is we want motifs to be in the center on 50 bases? Any guesses? It's fine if you have wild guesses. That's sorry? The weights at the ends will be mostly 0. Yeah, yeah. Sure, yeah, that's great. And what do you expect in the center, probably? Yeah, right. So that's great, right? That's kind of what you're expecting, because you find the motifs, but we want them to be in the center. So towards the flanks, 0 or something less than 0 is perfectly reasonable, and you only want to count them in the center, right? So let's actually do that. So our models have learned, and we can actually run these cells and just visualize the weights learned by these models. Actually, it's a very good guess, right? As you can see, towards the flanks, the weights that have been learned are close to 0 or slightly negative. Perfect. And in the center, the weights are pretty high, right? In fact, in the center, 1, 2, 3, and 4, and maybe like 4.5, 5 positions. And you can see each bin has a width of 35, right? And if you take 4 of those bins, that's 140 bases. And that's about the region which we considered when we inserted our motifs, right? So again, we didn't tell the model any of this, right? We just gave it trained examples. And those motifs could be anywhere, if you remember. They don't have to be at the 51st position or at the 32nd position or the 504th position, right? They are inserted randomly into the sequence. And the label is only determined by when you have an instance of the motif in the central 150 bases. And now if you look at the learned motif, right? If you remember, it was initialized pretty randomly, but you can now see that it's quite nicely picked up. So again, it's not looking like the PWM, which is a probability matrix, And there's no constraints. So as Rahul expected, we see a lot of negatives as well. But it's very clear that it's learned the CCAG, CTG portion of the motif. And in fact, there's even I guess it's not learned this part really well. So it's quite cool because we didn't really tell the model anything about what a motif is or what even position dependence in our labels is, but it was able to just piece together this convolution layer with the linear layer that it learned and just figure out this combination of functions that helps us discriminate between a positive and negative set. So comments, thoughts, questions? I'm just going to wait so people can sort of digest it. And please feel free to ask even other simplifying questions from the previous few cells. Yeah. Yeah. Yes. Like this is the architecture that we actually train for this example. If you go back to our lectures in the morning, people are asking whether we tell the model what these motifs are, but as you can see, we initialize them randomly. All we give the model as input are the examples, so sequences and their labels, plus or minus, right? And in the simulated example, yes, there was a pattern because we had inserted instances of this TAL1 motif, and then that's used that and their positional and where they were inserted positionally to determine the labels. And just based on that information, the model was able to figure out what the relevant motif is and where it needs to be for the examples to be, for the labels to be positive. The logo, right? Yeah. So yeah, maybe just to simplify what's going on here is all the values are coming from this matrix, right? And depending on so each column is a position. So we'll have 10 positions, right? So there's 1, 2, 3, 4, 5, 6, 7. So we specified a width of 10 for these motifs. And essentially, if we see a high C on the first position, right? That'll just correspond to like a 0.7 value, for example. And you see positive values for, I think, all of them here. So you'll see smaller positive values for G, C, and so on. At the second position, you see strong negative values for A, G, and T. So it's just going to have maybe like a minus 0.5. Why is because the scale really isn't important, right? It doesn't mean anything exactly because you can, if you scale something up here, you can scale it down in the linear layer. So it's all relative to. Yes, correct. It's consistent across the columns, but the absolute numbers don't matter here.
Sum across each of these columns.
They don't have to be. There's no such constraint. We don't impose these constraints. Right. Yeah. So I mean, the second column is telling you that if you see an AG or T, there can't be. I mean, that's not preferred at all, right? Yes. Yeah. No, I think there is a valid point where, at least in terms of the motif you inserted, both the C's have a high score, right? And that's not necessarily reflected in this. So I think that can definitely be some issue with the model. Neither, I would say. It's just some motif match score. Yeah, that doesn't necessarily have a physical meaning. The only like actual meaning you can attribute to is the final output, which is a probability of this, sequence being labeled as plus. So, so we have been able to show that you can correlate these scores with binding affinities from a separate assay. So there is some correlation, although you'll have to rescale these values. Yeah. Yeah. Right. Yeah. Right. I see. Yeah. Yeah. I think so. Yeah. I mean, I would, that would be my guess. But clearly, slight differences in the initialization also end up changing this. You're right. Yeah. 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 Mm-hmm. So let's see, that's the 9th column, right? This one? Yeah, I guess except the C and T are penalized less. So I guess, yeah, what Rahul is saying is you can just have an offset, right? You can perhaps just add a plus 0.2 to all the values here. It won't make a difference because in the end also you do have a bias term, right? So there is some flexibility where you can add a +2 here, a 0.2 here, but then remove from the final bias parameter the same amount. The interpretation there is that you can basically add or subtract a value from all the numbers in a column without really changing the meaning much. Yeah, I mean that's yeah, it's good things to think about because often we want to make sense of what the model is learning, and of course there's no constraints, and so as if you rerun the same thing twice, you're seeing slightly different versions. And in fact, I think you can just add say 5 to each of these and then get the same performance and the same exact model if you set 5 somewhere else. Yeah, so this model will not capture any interposition dependencies like the DWM. But now once you start stacking more layers, more convolution value layers, now then the model is less constrained and it can start learning positional dependencies between motifs. So if you start just adding more layers, right? So each, then I mean there'll be no constraint on each filter to actually learn a motif. So often it'll just be learning sub motifs and so on. And so there, I don't think you can parameterize it as a mixture model necessarily, just as a more flexible sort of nonlinear model. Yeah, I think I mean, this is sort of the meat of this tutorial. We have sort of a bigger model which has multiple filters. So again, it's a single convolution layer, but instead of learning one filter, we'll end up learning, in the second case, multiple convolution filters. So there's that's the second part of the notebook. Yes, so it's learning multiple motifs. It may or may not learn the tal motif in one filter, because again, it's not constrained to do so. I think in this case, it still ends up learning the tal motif in one filter. But I imagine if you rerun it a few times, you'll see that it's sometimes split across. Yeah, I mean, this simulation framework is called SimDNA that we used. It's very flexible. You can choose what instances of motifs you want to insert, where you want to insert them. how many you want to insert, where you want to exactly in terms of position, you want a normal distribution centered in the center. Yeah, for genomics, it's yeah, motifs, like inserting motifs, yeah. So yeah, what we can do is actually run the next few cells. So just to compare this with what we were doing before, the only thing that's really changed in this model is that the architecture stays the same. So the order of layers stays the same. All we've done is we've added a number of this number 15 here, which suggests that we're instead of using just this one convolution layer, we're going to use 15 of them. You can see we'll actually use 15 convolution filters. And so what that'll mean is that at the end of the first convolution layer block, instead of having one vector of values, we'll have a matrix of values, which will have 15 rows. And then we'll have the same number of positions. And so instead of having this vector output, we'll now have a matrix. And the model has more capacity, right? Because we're giving it 15 filters. So the number of parameters from 40 increases by 15 times for that layer. So this model has sort of more capacity. So in theory, you would expect it to perform slightly better. I actually don't remember if that happens. You can see. So yeah, we can run the remaining blocks. I'm just going to run through the end and see how everything looks. But as we increase the size of the model, you should also expect the training to take more time, right? Because there's more parameters, so you have to compute gradients for more parameters, and it'll scale linearly, usually, the number of parameters. And so you can see here, this is showing you the total count of models. This was 72 previously for the training parameters, but now by just increasing the number of layers by 15, we've increased the number of parameters to about 1,000. So. And again, we can compile the model with this, with the Adam optimizer and the binary cross entropy loss, use the same metrics. And once we set it train, you can see it's actually beating the simple model we had trained before, right? In fact, the accuracy for this model is slightly higher on the validation set, about 91.3% versus 94% on the training set. You can see the final accuracy is, I think, 90.96, which is not much higher than the single filter model we had learned. So in this case, adding all this capacity to our model didn't necessarily help us. It slowed down training, but I think the performance only increased from like 90.2 to 90.9. And that's mostly because in our case, the simple model was sufficient. It was the right model for our task in some sense. And so that was the right, it was a reasonable solution. And in fact, it did converge to give you reasonably high accuracy. And so you can see here again, there's a different loss function. If you remember, this looks different from our previous loss function. In the previous case, the model took some warm-up time really before the loss started converging. For this model with more parameters, you can see interestingly that from the get-go, the loss went down smoothly, the training loss especially. And you can see, of course, the validation loss, not as smooth. There's some bumps up and down, but on the whole, went down, eventually saturated. And that's where we stopped training. So previously, we stopped training at around 50 epochs for the first model. In this case, slightly earlier. So the loss seemed to have converged. Model seems to have converged much earlier. And again, we can repeat the process. And here you can see it's slightly more interesting, right? So instead of learning positive weights, for all of the ones in the middle, it's only learning that for some filters and not for all other filters. And actually what's going on is that since we have 15 convolution layers, even after the ReLU, there's a lot more values. So right, so there's actually 15 times more values after the ReLU as well. So we have a 15 by 30 matrix. And so each of these rows corresponds to the match for the different motifs that the model has learned. And then it tries to figure out the importance of each of these motifs, the final prediction. So you can imagine, since we've given it 15 motifs, and really what we know from our simulation is that only one motif is important, it's likely ignoring most of the motifs. And that's why, in contrast to the previous example where we had all high scores in the middle, this one has low scores for at least some of the motifs, but it has high scores for some others. And if you look at the motifs the model has learned, now it's learned 15 motifs, right? Because we set the filter size to 15. And so you can see a lot of these motifs don't necessarily look like the TAL1 motif that we inserted, but I think one of these does. So this one does look a lot more like the TAL motif. A lot of these are probably just ignored, right? So you can see that these weights, most of these weights, even in the middle, are close to 0 for a lot of motifs. And those likely correspond to some of these motifs. So again, we initialize all these motifs randomly. Some of them end up converging to be more like the TAL motif. The others don't, and I think the model just ignores them in this case, just associates them with the weight of 0. So in some sense, it's like we're not using the entire capacity of the model because the model is over-parameterized, at least in this case. But you can see it does learn the TAL motif, at least in this one example. So I think we're pretty much out of time, and we're also done with this notebook. As I mentioned, there's a lot of simulations available. Just to answer Rahul's question, so there's a bunch of simulations you can actually play around with in this notebook itself. It allows you to embed multiple motifs and see whether the model learns these motifs. Yeah, you can add things like grammar, where the prediction depends on the spacing between motifs. So it's like positive when the two motifs are close to each other, negative when they're farther away. And all these things do actually come up in real data as well. This is really how how real genomic enhancers behave. It often depends on the identity of the sequence elements, their characteristics, how many of them are present, the spacing between them, the orientation as well. But yeah, I think that's really it. So if anyone has any high-level questions or any specific questions as well, we can end with those. Yeah. Yeah. So the interesting about GKM-SVM is that it just schema counts, and it doesn't take into account positional information, right? Yeah. Yeah. Correct. So if you do one motif versus no motifs, it'll give you comparable accuracy to, yeah. I think maybe it'll be almost the same, if maybe slightly, noisy, but slightly better. Even motif counting, I think it'll be probably very similar. But once you start playing with position, where you're looking at spacing between motifs or absolute positioning, like in our simulation, where they need to be in the center, unless you make modifications to GKM SVM that take into account position, I don't think it will. Yeah. So absolute positioning, never that important, but relative positioning everywhere. I think for most enhancers, there are things like nucleosome-mediated cooperativity, where the accessibility or the activity is much closer when motifs are closer to each other and decays. For some of them, there's some interesting behavior where up to a certain point, the activity increases, but if you bring them further closer, then it starts decreasing. So yeah, so in those cases, we don't even train binary models. We just train regression models. And the readout is some notion of activity. It can be chromatin accessibility. So there's no real positives and negatives there. It's just, it's just a continuous value. So it'll tell you sort of how the overall activity changes as a function of distance between two motifs. Yeah. Correct. Yeah. 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 Correct. Yeah. 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 For sure. For sure. Yeah. Yeah. So CDCF will do pretty comparable because CDCF is just, yeah, it's kind of easy because it's really just the motif. This becomes interesting because for certain TFs, it's not just one motif. Often they have co-binding partners and other TFs that also enhance their binding. So in those cases, if it relies on another TF, then you're not just learning one motif. In fact, we've trained ChIP-seq models, people in the lab, for all the ENCODE experiments. And on average, they see for any individual TF, you see at least motifs of 3 to 4 transcription factors, because TFX that you're chipping or measuring can depend on the presence of the motif on another TF, also in a position-dependent way. So for those kinds of examples, you can't just do one PWM scan because there are multiple motifs that are involved in the binding of that one TF. So right. Yeah. So generally, so again, then you can get a lot of false positives because in general, not all motifs will be relevant. So if you take into account like 100 unique motifs even, the model may not converge to like a solution where it really only depends on the correct motifs. But I'm sure like you can probably work out some models that closely mirror the underlying biologies that are not neural networks, and those can predict reasonably closely. It's just in general, we don't know how many motifs to expect, what motifs to expect, and it's yeah. So if it repeats, but it is not predictive of your output, the model will probably not pick it up. Sometimes we do see some repeats, but those can really be a function of the sequencing experiment itself, biases, like sequencing biases. So in general, we've not seen a lot of false positive motifs. There is some of my recent work, which is looking at spurious motifs these models learn. But in general, if you're doing a single task model, it learns the right set of motifs.